Bienvenidos. Gracias por su asistencia. Somos los que somos, así que afortunadamente quien nos va a superar la cantidad. Les agradezco por su presencia. También a las personas que se juntan, que se presentan a través del canal de Zoom y el canal de YouTube. Es un gusto para mí presentarles a James Noble. James Noble trabaja en el National Physics Laboratory, Laboratory de Reino Unido. James hizo su doctorado en bioquímica en el Imperial College of London y se ha dedicado a hacer investigación sobre biosensores, y su trabajo se ha destacado mucho en el sentido de que ha tenido... Qué bueno que bien, persiguió una hoja con todos mis datos y la dejé en mi oficina. Pero tienen varios trabajos bien interesantes, por un lado, en la caracterización tanto de biosensores aplicados al COVID. En otro lado, también la cuantificación de marcadores específicos, como por ejemplo troponina, ¿verdad? ¿Me comentaste? Troponin, sí. Que es un marcador justamente de infarto cardíaco, entonces los niveles de marcador a nivel sanguíneo. Y un problema que también me llamó mucho la atención, con el cual uno se topa constantemente, es la correcta determinación de la concentración de proteínas. Cosa que, como todos ustedes han hecho alguna vez, se dan cuenta que no es tan fácil como uno dice, ¿verdad? Cuando tú ves entre lo que dijiste que tenías de proteína y lo que corriste en el gel, la diferencia suele ser grande. Y ahora James va a poner el día un poco con su nuevo trabajo, así que James, thank you very much for coming over, and thanks for agreeing to give this talk, and you're very welcome here. No tenemos el proyecto.
Estamos preocupados con lo de esto. El HMI conectado hay que prenderlo nomás.
Sí, ya lo perdí.
Ya.
Gracias.
¿Vale? Tiene una lucecita azul ahí.
Sí, sí.
Ahí está ya. Preocupados. Ahí viene.
Yes. Excel, PowerPoint, no.
No, es que está en Zoom, tiene que ir a un poco.
Perfecto. Which is your computer?
Es mi más.
Okay, no, so that's this. So that's not correct. What you have? It's still...
No, el otro computador. Está conectado al pasado de diapositiva al computador gris. Pero no está teniendo, ¿no?
You have that too.
Ahí lo ven, sí.
Okay.
Okay, gracias. Buenas tardes. So, thank you for the introduction. So my presentation today is on the design and development of self-assembled capsules for oligonucleotide drug delivery. So, as indicated in the introduction, I'm from the National Physical Laboratory. So this was opened in 1902, slightly different than 2002. So we have a remit basically to effect a union between science and commerce. So it's to ensure that the latest science is effectively and practically transferred to industrial and commercial life, and to try and break down the barrier between theory and practice. So what this means is that NPL, or the National Physical Laboratory, is the National Measurement Institute of the United Kingdom. So what we do is that we ensure we're there to support trade and industry through better measurement. So we hold a lot of standards that, for example, measurements are made to. So we hold the standard for, we do quite a lot of work on the standard for time. So what this means is that we can accurately measure time down to, I think, 10 to the minus 12 seconds. So this is important in telecommunications. So we hold a lot of the standards that basically other people make their measurements from, to ensure that the correct measurements are made and there's a traceability chain to these primary standards. What we also do is we do a lot of work around measurement science, trying to improve making more accurate measurements with lower uncertainty. So basically, industry has more confidence in the data that's being generated. And we do quite a lot of research in terms of improving these measurements, developing new measurements where there are challenges that industry face. For example, in production of say the next generation virus-like particles, what we try to do is we try to develop methods to help companies better validate and characterize their materials to get the products to market faster. So in this way, we're kind of supporting UK industry and also a European and worldwide level. We also work with NMIs, or National Measurement Institutes, throughout the world to ensure that the measurements are consistent, say in the UK, in China, in the USA, so everyone's measuring the same thing. So if you went for a troponin test, if you suffer from a heart attack, you get the same measurement in one country compared to another country, or even within the same hospital. So that's kind of gives a background of what NPL is associated with. The presentation today, I'm talking about basically we are trying to develop standards to support oligonucleotide drug development and more specifically delivery into cells. So to indicate, we are the NPL, so we're based in southwest London, about 15 miles away from the centre of London. We have, as an institute, 750 staff. We have about 200 visiting researchers per year. And we partner with over 200 organisations and 80 universities. So a lot of the universities are UK-based universities. We have PhD students with a lot of these universities, or we have active collaborations, and we publish with some quite high impact or high ranking universities. We work with lots of organisations. So within my group, we work with at least 40 different organisations. Often these are companies, and we're trying to support the companies in their measurement or in their measurement challenges. For example, that I gave for companies producing virus-like particles, we will help with their characterization of that. So yeah, a lot of the work and the research that we do is very collaborative, and it has input from university and also the commercial sector. So within NPL, there's lots of different areas of science. So there's a lot of engineering and physical sciences. There's a certain amount of environmental analysis, and say this is more physical analysis. And we've recently opened up a data metrology department. So I'm from the biometrology department, and typically we use physical methods to characterize biological samples or answer biological questions. So the majority of the group in biometrology is typically biologists, biochemists, but we have some people department or from chemistry departments who do a lot of work on the instrumentation side of things. So we can use, and we collaborate with groups across NPR, for example, mass spec imaging and surface technology, where they can provide some more physical methods to characterize our biological questions or our biological problems. So I'm in the biometrology group, and what we do is we develop materials and protocols to support standards, materials and protocols to support current measurement challenges within UK industry. So we often have national challenges. So, for example, antimicrobial resistance is the current one, and also vaccines and COVID-19 is another. So the government indicates the areas of research that we should be providing solutions for. And what we attempt to do is we want to generate a toolbox of standard reference material that people can then take. So this is basically materials that are very well characterized, and then they can compare them to their materials, and they've got a standard, so they can basically better characterize their material and have something to compare in terms of activity, how well their material is working. So what this means is that when I mean by better characterization, this is just an example of where we have, we're analyzing a cell. So we can analyze the cell at a chemical level with some of our colleagues. So they use mass spec imaging and Raman spectroscopy to give a chemical analysis of the cell. Then within our group, we will look at live imaging and kinetics, for example, using fluorescence, imaging of the cell, live AFM imaging of cells, including bacteria. We also collaborate with other groups that do surface acoustic imaging or measurements of cells. And then at the higher resolution, we tend to use transmission electron microscopy or cryo-TEM on cell slices to get a really detailed resolution of around a nanometer to try and get as much information as possible. So we will use multiple techniques to try and solve challenges or issues associated with biology. So this can include sample preparation, cross-comparison and validation of methods with other NMIs and also industry, instrument calibration and traceability, and also data integration, fusion and analysis, which with our basically big data team. So a lot of these methods that we use generate lots and lots of data, and one of the most important aspects is being able to derive the data that you want, all the answers that you want from complex data systems from these images. So the talk is basically centered around using self-assembly. So using very simple systems that self-assemble into more complex systems and using these as standards and imparting biological function into these systems. So this is basically how nature works in terms of you have more structural complexity and typically you have more functional systems. So nature starts with it, and we start with a peptide sequence that then folds, and you get secondary structures, for example, alpha helices or beta sheets, and then these systems with more functional systems or with more functionality. So, for example, these secondary systems assemble through often interactions between alpha helices and beta sheets into microtubules, for example, collagen or viruses. And then these can impart function into these materials, for example, cell motility, intracellular transport, mechanical strength, cell adhesion, migration, proliferation, angiogenesis, tissue morphogenesis and repair, and also cell transfection, for example, with viruses. So I will be mainly focusing on cell transfection, which is obviously the delivery of DNA into a cell, and then the subsequent biological effect of that DNA within the cell. But all we're doing is that we are taking what nature basically uses, and we're adapting these sequences to try and generate new structures and new functionalities through self-assembly. So self-assembly, people have been using self-assembly and what nature has given us quite some time. So there's a lot of work around hydrogels, and there's a lot of commercial products around these hydrogels using self-assembly from simpler monomer units. Then people have also developed fibers and tubes, and they've also developed particles and cages. So I kind of highlighted these capsules here in terms of the work that we're doing. We're trying to develop new capsules with new biological functionality. But this space is already, within this space, there's already quite a lot of commercial products that are used for transfection, for example, lipofectamine and Enter, which are known transfection agents that people use to deliver DNA into cells. What we're trying to do is we're trying to have this process more controlled and use self-assembly to develop new particles and cages that can be used as standards. So our aim is basically to mimic the functional complexity of nature by engineering peptides that we can self-assemble into complex morphologies. Again, the example here is fibra matrices, where we'll use fibers to generate 2 and 3D gels for tissue culture and tissue engineering. Or in my presentation, I'll talk about virus-like particles that can be used in gene therapy to deliver DNA into a cell. So what we want to do is we achieve this by having an improved understanding of the rules of peptide self-assembly to establish a link between drug design principles and biological function. So we're taking what nature does, adapting it, and then basically adding new functionality to these self-assembled systems. So the two main architectures that we use are the alpha helical coil. So this is shown here. So this is 2 alpha helices. What we do is we engineer one face of these alpha helices to be hydrophobic, and then the rest is basically charged. So these 2 hydrophobic faces basically interact with each other, and they can form basically more complex structures. So typically we use this for fibers and capsules. And we also use beta sheets. So we can modify peptide sequences to promote basically beta sheet folding. Then these form sheet-like structures, and it allows basically interactions at a tertiary and quaternary level. So we can develop more complex systems. So we'll be doing this. So there's a lot of potential in terms of oligonucleotide therapies at the moment. So this is just a brief list of therapies employing oligonucleotides or plasmid DNA. So for all of these, you need to be able to get the oligonucleotide or DNA into the cell, and it needs to be acted upon when it's in the cell. So for example, vaccines, expression of antigen hosts. So we'll see the mRNA vaccine from Pfizer for COVID-19 as a prime example of that. There's gene therapy where gene editing, for example, CRISPR, where you're modifying the current DNA that's in the cell. Gene regulation, so this, for example, could be knocked down using siRNA or antisense short oligonucleotides. And then the cell therapy, where you're reprogramming cells, for example, CRISPR. So all of these require the introduction of DNA, plasmid DNA, or short oligonucleotides into a cell. So some of the challenges are specificity. Ideally, you want to get the correct DNA into the correct cell type. Immune response. So obviously viruses are very good at doing this job, but the challenges with viruses are that, for example, adeno-associated virus, a lot of the population will develop an immune reaction against this treatment, which limits its applicability. Manufacturing scale-up. So there's a lot of challenges in producing virus-like particles and adeno-associated viruses at a large scale. There's also cargo restrictions with viruses. So, for example, adeno-associated virus, I believe, can only transfect plasmid sizes from about 4.2 to 4.6 kilobase pairs. So a lot of people and a lot of therapies, they want to use larger plasmids or get multiple plasmids into a cell at the same time. So there's a limitation on that. There's issues with toxicity with a lot of the peptide, chemical, or lipid nanoparticles that are used to deliver DNA into cells. And then there's also the issue of incorrect insertion of plasmids or DNA into the host DNA itself. So we believe that the use of capsules will be able to, self-assembled capsules will get around some of these challenges that face the oligonucleotide therapy area. So this slide just shows some of the metrology challenges. So we're interested in producing standards, but we're also interested in characterizing how well transfection works and characterizing the actual transfection vectors that people use. So that can be capsules, it can be virus-like particles, or it can be lipid nanoparticles. So associated with the vector, there's issues of size, formulation, and stability. So we're versus empty viruses or cap. And then, rather than going from analysing the actual drug itself, the mechanism and entry of release viruses, they typically end up in the endosome, but they have methods to basically release their cargo within the endosome. But a lot of this is quite challenging to replicate in virus-like particles or capsules to try and basically copy what viruses. This is the issue of intracellular location and targeting. So if you're trying to deliver a plasmid into a cell, ideally you want to get it into the nucleus. Sense oligonucleotides getting into the cytoplasm is fine. So there's certain basically targeting associated within the cell. And then there's the activity itself. Basically, you want to measure the transfection. You want to be able to measure the transfection efficiency and compare that to other vectors that you may want to use. And you also want to be able to measure the toxicity, because some of these vectors are toxic to the cells. You're adding a large amount of DNA into a cell. The cell generally doesn't like that and will respond. And also some of these agents basically punch holes into the membrane, disrupt the membrane to get the DNA into the cell, and this can be toxic. So our approach is to try engineer or transfer biofunctionality onto nanoscale capsule scaffold. So we've developed about, at the moment, 5 published transfer biofunctionality onto this scaffold and show biological effect. So shown here is basically we've infected macrophages with E. coli, and then we've treated them with our capsules that have a antimicrobial motif, and we can show using TEM that we can basically kill these E. coli, as an example of dormant bacteria in cells, using these capsules. And the concentration of capsules that's required to do this is a lot less than, say, standard antibiotics. So what we've done is we design capsules based on viral particle self-assembly rules, which are shown here for a viral particle, in terms of that you have basically simple protein-based self-assembled units that assemble together. So basically a lot of these viruses are basically 12 flat triangular faces and 12 vertices. So for a virus, this basically assembles within a cell using known assembly rules. And we're trying to do something similar in terms of forming similar kind of structures that hopefully will self-assemble into larger capsules that are very like viruses. So this is just a slide that talks about transfection agents. So typically transfection agents can be, so by transfection agents, I mean materials that you add to the DNA to get it into the cell. So typically these are cationic peptides. Typically these are cationic peptides, polymers or lipids. So for example, the Pfizer vaccine, mRNA vaccine, used lipid nanoparticles to get that into the cells. We tend to use peptides. You can use viruses or virus-like particles, or you can also modify short strands of DNA or RNA to make them basically to increase the uptake into the cell. So just shown here is basically plasmid, a TEM of plasmid DNA. It's partially condensed. So you can condense this with peptides or with polymers or lipids. So basically it makes the DNA a lot smaller and it gives it a positive charge, so it's taken up by the cell. What viruses do is they partially condense and then encapsulate the DNA from within the cell to then transfect to the cells. So shown here is a load of viral particles, and you can see most of them have got DNA on the inside from the staining. But what we're trying to do is we're developing capsules that basically self-assemble around the DNA, so you get condensation and encapsulation at the same time. These are some of the capsules shown here. These are about 20 to 50 nanometer structures that basically encapsulate the DNA and then self-assemble around it. So why use self-assembled capsules as standards to assess transfection efficiency? So the reason we're doing this is it allows comparison of transfection experiments, or the analysis or the comparison of new agents. So people can basically take these standards and they can assess how well their transfection is working or how well their new transfection agent is working compared to our standard. And it gives people greater confidence in the transfection measurements. And with some of the supporting measurements that we're doing to better characterize transfection, we're hopefully turning what is a qualitative kind of technique to a more quantitative technique. So we believe that in using peptides that self-assemble into capsules, the advantage of these is that they are chemically pure, so peptides are very easy to analyze. We know that the impurities that are present in there, they're very simple systems. They're very easy to assemble, so we can basically just solubilize the peptide in buffer, and within 30 minutes form a capsule that then people can use straight away to validate their methods. They're safe, so you don't need special labs because you're just using peptides as opposed to, say, viruses that may require Cat2 or higher facilities. They're very easy to modify, so we can modify the surface of the capsules to impart additional biofunction, and also the scale of production. So we can use standard peptide synthesis to produce these capsules. So it'll take maybe 4 days to synthesize, and we'll get a yield of anywhere between 20 to 100 micrograms, milligrams, and this can be scaled up. So if you imagine VLP production or adeno-associated virus production, this can be quite complex. It can take a long time to achieve because you need to grow certain cell lines. There's lots of purification steps. So we believe that these standards are more applicable, that more people can use them without specialist facilities. And there's also a traceability chain. So we know the exact concentration of the peptide, so we can measure uptake into the cells with a very high degree of accuracy because there's a traceability chain to the mass of this basically peptide that is available as a standard. So we've developed, or we've published about 5 systems that form capsules. So 4 of them use peptides to self-assemble into larger capsules. And then we've got one that basically uses DNA origami to form a box. So each one uses a different motif, and we tend to generate different sizes and different properties of these capsules. One we currently have is TechVir, which is around about 12 nanometers. Then we've got about 3 of them that are around about 20 nanometers, which is our C3 capsid and D capsid. So the L and D capsid are it's the same motif, but we're using L amino acids and D amino acids. So D amino acids are more stable to protease degradation, last longer in circulation, and they may be more stable within the actual cell itself. So these are all around 20 nanometers, including our DNA box. And then the L capsid capsules they tend to be they generate more heterogeneous size particles. So we tend to have particles around 20 nanometers up to about 200 nanometers for our L capsid. So we've got a range of capsules that we can use depending on the function and the requirement or what oligonucleotide we need delivered. So the first one that we published was capsid. So this uses a beta sheet motif. So these tend to generate larger capsules that are heterogeneous in size, as shown by the TN here. So we've got small capsules around about 10 to 20 nanometers, and larger ones up to 200 nanometers, which is not ideal. But what we did here is we took the native sequence of an antimicrobial protein. This here is the beta sheet part. We removed this, we engineered it, and then we basically allowed it to form a triskeleton where we've got 3 beta sheets sequences that are joined in the center as a triskeleton format. So this basically triangle is quite similar to what I showed previously as how viruses self-assemble. And then these beta sheets, basically this interacts with another section, an adjacent section of this to form a much larger structure, which is basically, which then forms a capsule. The reason why we know they're capsules is that we've used cryo scanning electron microscopy, and we can see lumens within these particles, which indicate that they are capsules as opposed to solid nanoparticles. Then we've done more characterization with atomic force microscopy, but we're also interested in basically the biological function. So we've used an antimicrobial sequence, so we know that these are basically, the capsules have antimicrobial properties, but they can also be used to deliver siRNA into cells. So here we labeled the siRNA with a fluorophore, and then we added them to GFP expressing cells. And as you can see, we get uptake within about 24 hours or less using capsid. And it's the uptake levels in terms of getting siRNA into the cell are similar to that of lipofectamine, which is a well-known commercial transfection agent. So we know that we can uptake siRNA into a cell. We can also show bioactivity of that siRNA in terms of we measured the mRNA knockdown. So we compared our capsules, the efficiency in these cells. So we showed basically knockdown using various formulations of our capsid, and they were approaching the activity of that of lipofectamine, which is a commercial agent. But this was our first attempt at forming capsules for this purpose. So we were hoping to basically improve on the efficiency of knockdown, as in improving the biological function by optimizing the capsule itself. So our second capsule is called TechVir. So this uses an alternative approach, which is an alpha helical coil. So from a previous slide, you have two coils that basically interact with each other. If you modify the sequences of them, you can change the orientation of how they interact with each other. And our molecular modeling shows here basically how they form an empty shell-based structure. And then this just shows the filled capsule with sRNA on the inside. So here we just show that basically on the TEM, it forms particles that are about 12 nanometers in diameter, but you can see that we've got basically a dark staining. So on the outside is the shell, and this is the lumen on the inside. So you can see this basically at a large scale of basically the scale bar there is 100 nanometers. So we're producing lots of capsules. And as we magnify and go closer in, we show that we are producing capsules with the lumen on the inside. And this is just a phase contrast of this TM image to show that the lumen better. And then what we did is we used cryo-EM tomography, where you're taking basically cryo images of the capsule at various different planes. And then you can reconstruct those images. And we reconstructed them to show that we basically had formed a capsule with a lumen on the inside. And then this was modeled based on HIV virus, which is a lot larger. I think this is about 80 to 100 nanometers, but we are forming similar structures to basically viruses, as shown here through self-assembly. So then the third model that we looked at was again an alpha helical coil. So these tended to form small homogeneous capsules in terms of the size. So this is shown here in the TEM, where we're getting about capsules of 20 nanometers. We're getting lots of capsules formed. So the approach that we took is that we wanted to form this triangular motif that's seen in a lot of viruses. So again, we're using a triskeleton. So the blue peptides are joined together in the center, and these are cationic. But then in this system, we added a second peptide, which was anionic, which bound to this cationic peptide to form a coil. And then these had a cysteine at the end of it. So what we had is triangular structure with cysteines at the termini of the triangle. So these cysteines basically bound with cysteines from other of these triangular motifs to form this 5-star kind of ring structure. So within this ring structure, there's 5 of these peptide motifs. And then these form together to form the capsule itself. And this is shown basically in this schematic here, and just this from a different view from above. So it's not complete self-assembly because the size analysis shows that we get an average of around about 18 nanometers, but we've still got some of the subunits that haven't fully formed this capsule present in the sample. So we engineered this peptide to have antimicrobial effects. So what we found is that we tested these on bacterial membranes, and we measured the activity using atomic force microscopy. So this cationic peptide sequence here, we know disrupts bacterial membranes. So what it does is it inserts into the bacterial membrane and it disrupts a lot of bacterial functions, for example, electron transport across the bacterial membrane. And what this does is it tends to kill bacteria. And this can get around resistance because often bacteria don't have resistance mechanisms to stop this form of antibiotic from working. But what we found is that the capsule itself was a lot more efficient at killing bacteria than just having this cationic peptide sequence here. So basically incorporating this peptide sequence into a larger capsule structure made the antibiotic a lot more effective, i.e. it killed bacteria at a lot lower antibiotic concentrations. And just shown here is a trace of basically an trace where we are measuring the insertion of these monomer units from the capsule into the bacterial membrane. So we can see that basically on a basically bacterial membrane scaffold, we are forming, we're destructing the actual lipids, forming holes in the lipid membrane of about 2 nanometers deep, well, 2 to 4 nanometers deep, which represents just one of these arms of the monomer units that form the capsule.
James, sorry, just to be precise, then the figure we see in D, the two there, that's one way of looking at the capsule, and the other is looking at it from another angle.
Yeah, different plane.
And although they are quite homogeneous, they tend to form different structure, and it's the peptide that itself that will be antimicrobial, not what you're putting inside the capsule.
Yeah, so in this case, it's the peptide itself that's antimicrobial.
So when it meets a bacteria, then it will disassemble it and disrupt it. And
yes.
What gives it selectivity? How do you make it selective?
So the selectivity is within the peptide itself. So human membranes have a different lipid composition than bacterial membranes. So you can modify the peptide so it tends to favor bacterial membranes over the human membranes. So what we tend to do is we also measure a kind of therapeutic index where if we're treating the bacteria, well, if we're using these capsules, we'll test them on human red blood cells and bacterial cells, and ideally we want a therapeutic index of at least 1,000-fold preference for bacterial cells in terms of killing of them compared to red blood cells. So, but the specificity is engineered within the peptide sequence itself, but these capsules are a lot more efficient at killing the bacteria than just having the single peptide sequence on its own. And we've got videos to show this, but I've got in a next slide, in a future slide, we've got a series of AFM images that show basically the mode of action.
And could this become a 1-2 punch? I mean, encapsulating an antibiotic inside the system, so delivering the antibiotic directly to the bacteria, this would really take care of antibiotic resistance.
So theoretically, it would improve that because, you could incorporate 2 different methods to kill the bacteria. So you could either have an antibiotic within this capsule, if you could tailor its encapsulation, or you could also include an oligonucleotide that could be directed against the bacteria to basically modulate gene expression, gene function within the bacteria itself. So there is the potential to do that. We've not attempted that, but there is a potential to do that.
But would adding something inside the capsule actually alter the way the peptides are formed in the capsule?
So for some of our systems, yes, it does. For this one, we haven't tested it. We know on this one, so for Tecvir, when we add the sRNA, we get a slightly larger capsule, so it goes up from about 12 to 20 nanometers. But for most of our systems, you only see very small changes in size. The capsule still forms around the cargo. So basically we have a self-assembled scaffold system that we can basically tailor with different biological properties depending on the peptides that we use, or the motifs that we can add to that peptide scaffold to add additional biological function. So some of the things that we do is that we've got microbial action of capsids. So this is basically a live dead fluorescent staining assay. So the bacteria are alive when they fluoresce green, and when they fluoresce red, it indicates that they are dead, or we've basically orated the bacterial membrane and they're close to dying. So when we add capsids, we see a massive increase in the number of dead cells. We've got fluorescent techniques, super resolution techniques that allow us to look at the actual membrane itself of the bacteria. So we can understand how the capsule is affecting the integrity of the bacterial membrane. Again, this just shows transfection of fibroblast cells. So we're delivering a GFP expressing plasmid into the cell using our capsules. And after 24 hours and 48 hours, we see GFP expression. Converse to that, we can knock down GFP expression within cells using siRNA. So we deliver the siRNA using our capsules. So this is an example of basically cells where there's no siRNA added. And this is an example of where we're using capsules with siRNA, and we see a massive decrease in the number of cells expressing GFP and the levels of GFP expression. And then this is, again, another example of just being able to deliver fluorescently tagged siRNA into a cell. So this is our capsule. And after 6 hours, we see quite a lot of basically uptake of siRNA into the cell compared to lifefectamine, which is a typical siRNA transfection agent that's used in lots of experiments. So our capsules perform close to a lot of the perform levels close to a lot of transfection, a lot of the activity that a lot of basically commercial transfection agents work at.
James, just to follow up on the previous question.
Yeah.
So how specific can these peptides be? I mean, could these be engineered to be specific to, I don't know, Plasmodium falciparum membranes, for example, in order to tackle malaria? Or is it just a bacteria versus eukaryotic?
At the moment, it's just bacteria versus eukaryotic.
Okay.
Because we know that there's differences in the membrane that we can basically target. Theoretically, we could add targeting functions to the capsule on the outside to allow us to target different types of cells where there isn't differences in the lipids. So this is things that we're looking at the moment. I'm not saying we've been 100% successful because we may not be able to get complete display on the outside of the capsule, and it may disrupt the capsule formation. So this is what we're doing at the moment, but we are aware that there may be challenges associated with that. So adding a targeting modality is adding an additional biological function to the capsules that we've currently got. So it is kind of like a next step, and it could be quite challenging. So I've shown basically 3 of the 4 systems that use peptides. So we worked with UCL, University College London, University College in London, that does DNA origami. So we kind of did it, and we played around with the idea of doing a DNA-based virus, which is like an inside-out virus. So we used DNA origami to deliver a protein into a cell, whereas a virus is using basically proteins and lipids to deliver DNA into a cell. So we had basically a box scaffold here, and you have basically hinges that allow you to close the box. So in the absence of the hinge DNA sequence, we get basically it just looks like a bit of flat Lego on the atomic force microscopy image. When we added the hinge sequences that allow the box to form, we get basically these square boxes that are a lot smaller and that show an increase in the height. So we've managed to form boxes. So then the next trick that we did is that we engineered the hinge so that it was pH sensitive. So the box was stable at normal pH around about 7 to 8. But when the pH dropped to about pH 5, which is the pH of the endosome, the box opened up and it released its cargo. So we measured this using FRET, fluorescence resonance energy transfer. And then we also wanted to confirm basically if we could use these DNA boxes to deliver material to deliver GFP into a cell. So what we did is we tagged the box with HIV TAT sequence that was labeled with Cy3 fluorophore. And again, shown here, we can see uptake of the Cy3 TAT box. And then when it went to the cell, we could and released the GFP, within those cells, we could see basically GFP expression. Sorry, not expression, but basically the presence of GFP in these cells. So it's not the most efficient way of getting proteins into a cell, but if we just added GFP to the cell culture, or we added the box with GFP inside the cell culture, we saw a large increase in basically cells showing basically GFP uptake into the cell.
James, sorry, and this GFP stays within the endosome, or does the box open up and break up the endosome as many?
We assume that the GFP is staying in the endosome.
Oh, okay.
So we haven't engineered any functionality onto the DNA box to get it out of the endosome at the moment.
No, I was thinking that maybe just by opening it might burst it.
Possibly, but we just don't have any evidence of that at the moment. So we, unfortunately, when we publish this, we can't tell if the GFP is localized in the endosome or whether it's escaping the endosome. So yeah, it would have been nice to be able to prove that, but we could only prove basically release at lower pH.
How, like a DNA box sounds very unstable physiologically, like it's full of DNAs, like DNA enzymes. So how does it stay like how does it stay structurally sound enough to reach the endosome in the first place?
Okay, we don't know. So yeah, the DNA is so theoretically, DNA will be degraded when it gets into the cell. I mean, typically when it gets, I believe when it starts to get into the endosome, there'll be if you reject it into basically a say a human or an animal, there will be DNases that would degrade the DNA early on. So we know that this is purely for transfection into basically cells in culture, not into say human tissues or anything more complex that may have a lot of proteases. We don't know the long-term stability of these cages when we basically use them in cell culture. So some of this release may be associated with the degradation of the box, associated with DNases within the cell itself, if they're expressing it, or within the culture medium itself. But we're confident that enough of the box is stable enough to get into the endosome to basically enact the mechanism of release.
Right, like because I know, like at least in some cultures that we use DNases in the culture in order to prevent viruses from.
Yeah.
Okay, so we wouldn't be able to use those.
Absolutely. Yeah. I mean, these do have severe limitations. I mean, I guess I've shown this as just another example of a self-assembled system that we've developed, but we know its limitations for transfection, or not transfection, but delivering protein cargoes into cells. I think it was more to show that we can develop basically certain structures using DNA as well that we can impart biological function into that. I don't know enough about modifications of the DNA and the influence on what structures you can form So you can make the DNA more stable by chemically modifying it, but you're theoretically changing a lot of its properties, and it may not be possible to use to be able to form these DNA origami structures using this modified DNA. Yeah, it isn't probably the best if you want to deliver proteins into a cell. It may not be the best tool to use if you've got DNAs in your cell culture medium.
DNA origami is good enough. It's amazing enough.
Yeah, I mean, people are trying to use, so the group that basically, we don't do this, but the group LCN, they do a lot of work with DNA nanopores. So again, they're potentially using systems where there is DNA contamination could cause issues. And there are ways to make it more stable, but it may limit this kind of structures that you can generate. So we've developed these systems that we hope to be able to use as standards. We've also developed quite a few measurement tools, again, to support the characterization of our capsules, but also of virus-like particles and other transfection agents that people use. So they can have more confidence in the production and the purity of them. So we use a technique called fluorescent SIM. This is structured illumination microscopy.
Mm-hmm.
So this is a method that basically takes the images in multiple planes, and it allows you to reconstruct fluorescence images. So it's similar to confocal microscopy, but it allows you to reconstruct the fluorescent image, and it gives you a resolution of about 100 nanometers. So shown here is basically the killing of a bacteria using live dead stain with our capsids, and we can see basically quite detailed structures of the bacterial membrane and the bacterial architecture itself, and show disruption and changes in the actual bacteria shape. So we, with collaborators at using synchrotron facilities at Harwell, we've used small angles X-ray scattering to basically characterize our capsules to show that they have lumens inside. So we can show that they're spheres by using X-ray scattering. We can also show that they have got a lumen on the inside, as in they are capsules, not particles. So then we also use TEM and cryo-TEM to show our capsules in terms of measuring the size, whether they're filled or not, and also whether there are any defects, so they haven't formed perfect capsules. So we can get an idea of the purity of our capsules, whether there's basically how well they have formed capsules, or whether there's basically a lot of monomer or other structures present in the sample that indicates that maybe our capsule isn't the best one to use. Again, we can use optical spectroscopy in terms of here circular dichroism to show that our peptides or our capsules physically in cargo. So as shown here, we get a change in CD spectroscopy as we increase the concentration of siRNA. So then from this, we can basically show how well certain peptides interact or bind to these oligonucleotides. Again, with our collaborators at LCN, we can use liquid AFM so this is using AFM but in a liquid mode, so we can, in real time, we can add our capsules to a synthetic bacterial membrane and observe them. So the capsule here is shown in white, so we can observe it binding to the membrane. We can observe the capsule then inserting into the membrane and then breaking up and causing additional holes within the membrane itself. And then what we've done is, again, with collaborators at King's College London, is use molecular modeling to model this basically in the initial interaction of the capsule with the membrane, and then its insertion in the membrane and the formation of pores. So here we're trying to understand the reason why a capsule is more efficient at killing the antibiotic than, say, using just the peptide monomer. So We develop capsules, but we also develop supported measurement infrastructure to better characterize the systems that we're creating. So to conclude, we've developed, through self-assembly, we've developed peptide capsules of various sizes and properties. We've shown that we believe biofunctionality can be engineered into the capsule scaffold. So, for examples I've shown are oligonucleotide delivery into cells or antimicrobial activity. That these capsules have application as standards that people can use for oligonucleotide transfection. So to basically compare their transfection, users controls in transfection experiments, or to assess the relative activity of their own transfection agents. But at the moment, we've got challenges to try and improve the capsule bioactivity, so it's comparable with viruses. So obviously, viruses don't just deliver DNA to cells. They then allow the DNA to be transported into basically the nucleus. They allow endosomal escape. After basically the viruses are formed, they then facilitate basically the virus export from the cell. Viruses have a lot more functionality than we can currently engineer into our capsules. At the moment, we've basically maybe got like one or two different functions. We've managed to incorporate one or two different functions into our capsules. So the challenge is to maybe be able to increase the number of functionalities that we can incorporate into the capsule by basically adding them to the exterior of the scaffold to try and get the biological activity to be comparable to viruses. I mean, viruses have evolved over billions of years, so we are way behind in what we're trying to do. But if we can engineer more bioactivity into our scaffolds, it gives us better confidence that we can produce more useful capsules for transfection or for killing bacteria. So what I've shown is just a small part of what our group does with self-assembled structures. So we have about 20 scientists within our group, and other areas that we've explored using self-assembly include optogenetics. So we have an interest in being able to control gene expression in human cells or potentially in agriculture using optogenetics. So that is basically using light to control gene expression. But with these systems, it's quite challenging because often you have to introduce multiple genes into a cell to express multiple proteins to be able to control expression of genes, specific genes. And for viruses, this can be quite challenging in terms of there's different sizes of plasmids that you need to introduce, and there's multiple copies, or sorry, there's different types of plasmids that you need to introduce. So the capsules may be slightly better than using viruses in that we can tailor and that we can introduce plasmids of various sizes or different types of plasmids into the cell. So we're exploring the use of capsules in optogenetics. We're also interested in using the capsules as potential vaccines or standards for vaccines. So as we showed, we can produce a capsule of around 20 nanometers, and we can coat the surface of that capsule with antigens. So the hope is that we can basically use these as potential capsules for antigen display, and therefore they may be able to be used as vaccines or standards for vaccines. So we do quite a lot of work forming fibers as well. So the alpha helices that we use, we can basically create fibers that have a diameter of about 3 nanometers, but a length of between 100 to 1,000 nanometers. So what we've found is that we can reproducibly form these fibers, and when they're visualized under TEM or other imaging techniques, they form structures that have very defined length scales between the individual fibers that form these larger fibers. So we're using these as TEM standards for length measurements. So we found that we could basically, using these standards, we've developed standards that can be used on TM images to accurately measure length scales from about 0.3 to about 10 nanometers. So current standards that people use in TM analysis to make very accurate length measurements are typically at the 100 nanometer to 1000 nanometer scale, as in people are using gold nanoparticles. So we believe that our TM standards can allow people to make better measurements at the lower nanometer and sub-nanometer scale. And then we also work on cell scaffolds. So we use some of these fibers to form 2D and 3D gels. Again, these can be used as cell scaffolds to grow cells in culture, either 2D or 3D culture, or potentially they could be used to transfer cells or transplant cells into human patients in terms of tissue engineering. So we're doing quite a lot of work in terms of standardizing and preparing measurements for cell scaffolds. So we're using these self-assembled systems, the rules, to generate materials in these areas. So that's the end of the presentation. I'd just like to thank basically our funding, where we get the funding from, which is typically government from the Department of Business, Energy and Industrial Strategy, but we also get funding for some of this work from these funding bodies as well. And just to indicate that what we're trying, this is just a small part of the work that we're trying to do to develop reference materials and methods. And hopefully we'll have an open source toolbox of basically reference methods and protocols and reference materials that people can use for various biological activities, with the aim of being able to have more confidence in the data that they generated and have reference materials so they can make more quantitative measurements of their own systems using our reference materials. So that's the overall aim of the work. So that's the end of the presentation. I don't know if there's any additional questions.
I did have a question. Maybe I missed the first part of the presentation. Maybe you explained it already, but I'm very curious about the chemical structure of the crystalline structures you were mentioning.
Yeah. So I think the best one is probably, well,
yes, the alpha helical, the coils.
Yes.
Because it's you said it's is the whole, are the three cations peptides, how are they joined? Like, is it a peptide union, or is it some other sort of covalent union?
So
right there in the middle.
So this is like a monomer unit. So we have three peptides that are joined to form the triangle triskeleton. But then these are basically formed through the cysteine, so disulfide interaction.
The skeleton itself is just a monopeptide that has
So these blue sections are just a single peptide.
Oh, okay.
And then we have one of these, say, triskeleton peptides, and then we add three of these anionic peptides with the cysteines on them to form this triangular unit.
Larger structure.
But then we have so in another system, we don't have any cysteines in this system. We're just using the beta sheet, the density of beta sheets to basically interact with each other to form these more complex structures. But we believe this is one of our initial capsules that we developed. So in terms of using this beta sheets approach, it may generate more heterogeneity in the overall size of the particles, whereas using basically these triskeleton units with cysteine interactions to form this, we believe we may have better control over the heterogeneity of the size in terms of there's less basically heterogeneity associated with the size of these scaffolds. But we haven't tried it with enough systems to absolutely prove that.
Yeah, as a biochemist, the structure of bifurcating peptide is very, I don't know, I haven't seen anything like it before, so it's, I think it's very interesting and cool that how you develop that.
Yeah. Oh, thanks. Yeah, I mean, it's the entire group. So yeah, it's not just myself, but yeah, I mean, some people in our group have spent quite some time, maybe 15 years, playing with these self-assembled systems to see what kind of materials we can create.
More than a question, I have a commentary for the presentation. I was thinking that one of the principal challenges for to create a system to transfect eukaryotic cells like the inside the nucleus is that viruses for do that task along the capsid they have another structure that is called the portal vertex, which attach the nuclear core. So to design that structure, I mean, I think it's very difficult.
It could be very challenging. So I think we believe that we can add small, say, antigen sequences or peptide sequences to the outside of our capsules. Whether we could add larger proteins, we don't know. It is potential, but it could be quite challenging. But the issue with that would be more the, we believe these systems are quite straightforward because we can synthesize them on a peptide synthesizer. So we can basically just synthesize the entire sequence, put them into solution, and hopefully they fall into the correct self-assembled structure. If you're adding proteins to that, you're adding a whole additional complexity in terms of attaching the protein to the peptide scaffold itself, which it could be done, but it could be quite challenging. But there is ways that we could approach that in terms of attaching material to the capsule post assembly of the capsule. But again, there may be issues with efficiency and heterogeneity of the capsules created thereafter. But yeah, we know that especially for the DNA transfection, that one of the main challenges is getting the DNA into the nucleus, and there are other ways that could be addressed, but we haven't had a chance to play around and add that additional functionality to our capsules at the moment. But it is probably one of the reasons why they are not as efficient or as good as viral vectors.
¿Más preguntas, comentarios? James, I was thinking of what you mentioned about the measurement standards in electron microscopy. It's really notable because we're getting to really low levels of resolution, but with that resolution, we are also losing the capability to determine how small we're getting.
Mm-hmm.
And it's, I mean, I remember when I was taking classes, people would tell us, oh yeah, a red blood cell is 7 micrometers, and when you're in EM, you can see an actin filament, and it will be 7 nanometers. But when you're going below both limits, then how do you set the structure? You compare it with the AFM or?
So I did have a slide and I removed it because it's to basically show a lot of this development. So we are using, so TM is it's a useful technique, but you're not actually measuring the peptide or the material itself. You're actually having to stain the material. Mm-hmm. What you see is the stain. But we've used, I believe it's scanning tunnel microscopy and a few other techniques that I can't remember the names of, that don't require basically staining, to give us more confidence in the size of the fibers that we're generating, that we're using as standards. So this is why I believe that we're approaching basically 1 nanometer in terms of the resolution that we can see using these techniques. But I would have to, this isn't my work, this is someone else's work, so I have to confirm that. But yeah, it's trying to develop standards where there's an unmet need. At the moment, we believe there's an unmet need there as people push the cryo-TM techniques to the max. Hopefully, our standard is providing the material to have more confidence in some of the measurements that they're making, and especially length measurements.
Thank you. One question regarding the organization. Do you receive students or postdocs or PhDs that might be interested in working with you in these things?
So we've got various mechanisms to do that. So we have within the group, it varies, but we had pre-pandemic, we had about 10 PhD students. Funding PhD students can be challenging, but it can be very good. So this is typically how we work with universities like Oxford, King's College, UCL. We also have guest researchers that come over and will work in the lab and develop their skills and maybe pass on skills to us as well. I think at the moment we're trying to work out how all that works after COVID.
Yeah.
So it's, but yeah, in terms of visiting researchers, yeah, we do have quite a few of them.
Okay, thank you. So, thank you very much. So I'm gonna stop this.